Hello friends, in this video I am going to focus on first reaction of photosynthesis, that is light reaction. Okay, in my previous video I discussed about the concept of photosynthesis, the site where photosynthesis occur, that is chloroplast, and different pigments of the plant which is involved in the process of photosynthesis. In this video I am particularly focused on the first and important reaction of the photosynthesis, which is light reaction. Light reaction take place in the granum of a chloroplast, particularly in the membrane of the thylakoid, and this is the reaction which is responsible for absorbing the light energy and converting into the chemical energy in the form of reduced energy carrier like ATP and NADPH2. The product of the light reaction is used by the dark reaction for fixation of carbon dioxide. We know that dark reaction, which is also called a C3 cycle or a Kelvin cycle, require high amount of energy for production of single glucose molecule. Approximately 18 molecules of ATP and 12 molecule of NADPH is required by Kelvin cycle for synthesis of single molecule of glucose. So Kelvin cycle always have high energy requirement, and the energy requirement of the Kelvin cycle is fulfilled by light reaction. Okay, so in this video I'm going to discuss in detail about the light reaction, its type, and how exactly it occur inside the chloroplast. Okay, in my previous video I discussed about the structure of chloroplast. If this is a chloroplast, then it is a double membrane organ having outer membrane and inner membrane, and inside the inner membrane aqueous fluid substance is present, and that fluid substance is called as stroma. While green sac-like structures are present in the chloroplast, they are grana. Single sac of the grana we called as a thylakoid. Okay, so thylakoid. Actually, light reaction take place inside the chloroplast in thylakoid. Okay, so if you want to understand the actual mechanism of light reaction, you have to focus on the thylakoid structure. Okay, so I draw this thylakoid single sac here. This is the thylakoid. Thylakoid also having a membrane, phospholipid membrane, and the internal portion of the thylakoid is called lumen. The light reaction take place in the membrane of a thylakoid. Actually, thylakoid membrane having both the type of pigment which is present in the plant, like chlorophyll, which is an essential pigment, and carotenoids as accessory pigments. This pigments is present in the membrane of the thylakoid in the form of pigment system or photosystems. Okay, so the pigment is present here. If it is first or photosystem 2, first reaction center, which is called as photosystem 2, it is present in the membrane of the thylakoid, and then electron transport chain is present, which transfer electron from photosystem 2 to photosystem 1. This is PS1. And from photosystem 1, again electrons are transferred. Okay, and finally NADPH is produced in the light reaction. The path of the electron in the thylakoid membrane is look like Z, English letter Z. So the light reaction is also called as Z scheme. Okay, both the photosystem 2 and photosystem 1 absorb light of a different wavelength. Photosystem 2 absorb light of 680 nanometer maximum, while photosystem 1 absorb light of 700 nanometer. And when electron transfer from photosystem 2 to photosystem 1 by electron carriers, some hydrogen ions are pumped from stroma. Okay, remember that outside this is the thylakoid sac, and outside the thylakoid sac is a fluid film matrix which is called stroma. Okay, so when electron are transferred in the electron transport chain in the membrane of thylakoid, by using the transfer energy, some hydrogens were pumped from stroma to the lumen of the thylakoid. Okay, and due to pumping of the hydrogen ion concentration increased in the thylakoid, and due to which a hydrogen ion gradient is produced. We know that all biological gradients which is produced in mitochondria, bacterial cell membrane, or inside the lumen of the thylakoid have energy. And when this hydrogen again come back into the stroma, and they produce ATP. Okay, this hydrogen when accumulate in the lumen, they come back into the stroma through a special protein which is called as ATP synthase. And this ATP synthase use energy of hydrogen ions for conversion of ADP plus IP into ATP. Okay, this is the general view of light reaction. Okay, so at the end of light reaction, both NADPH and ATP is produced outside the thylakoid membrane in the stroma, and we know that dark reaction generally occur inside the stroma. So both these energy carriers is required in the stroma, so they produced outside the thylakoid directly in the stroma. Okay, so this is the basic idea of the light reaction. The process in which ATP is produced in the light reaction is called photophosphorylation. We know that the process of synthesis of ATP in living organism is called phosphorylation. There are various types of phosphorylation like substrate level phosphorylation, oxidative phosphorylation, and now here you can see photophosphorylation, the process in which ATP is produced by using light energy. That means ADP and IP are combined together to produce ATP in the presence of light energy. So this process of synthesis of ATP in the light reaction is called photophosphorylation. Dr. Arnold discovered two types of electron flow in the light reaction of the photosynthesis. In first flow, electron when leave from photosystem 2 after entering into the photosystem 1 and then into the NADPH, they do not come back to the photosystem 2. Okay, and that means one-way traffic is occur. So that type of electron flow is called non-cyclic photophosphorylation. Okay, and in cyclic photophosphorylation, electrons which leave from the photosystem 1 again recycle. Same electron come again into the photosystem 1, and that is called as cyclic photophosphorylation. So light reaction in the plant having two different types, non-cyclic electron flow or non-cyclic photophosphorylation and cyclic photophosphorylation. In my today's lecture, I am going to discuss in detail about the light reaction of the plant, then both the variation of the light reaction, that is non-cyclic photophosphorylation and cyclic photophosphorylation. Before starting with the light reaction, you should know few basic things like light reaction occur inside the chloroplast. Actually, it occurs in the thylakoid sac of the chloroplast in the membrane of the thylakoid sac. Okay, outside the thylakoid sac is the stroma, while inside region of the thylakoid sac is called lumen. Okay, once you clear these things, now we can start with the actual light reaction. Okay, so as I said earlier, light reaction have two different type of a photosystem, photosystem 2 and photosystem 1. These systems are nothing but the complex of the pigments, both accessory pigment and essential pigment. That means carotenoid and chlorophyll respectively. Okay, now this portion of the membrane of a thylakoid I enlarged here. Okay, for the understanding purpose, I have drawn this portion of the thylakoid membrane, okay, here. And now I'm going to show you how exactly photosystems are located in the membrane and how the electron flow take place. Okay, so first of all, this is photosystem 2. The main pigment of photosystem 2 is chlorophyll A. This pigment is called as reaction center because actual role of transferring light energy into the system is carried out by reaction center. Okay, this is chlorophyll A. Now this chlorophyll A also have some accessory pigments like carotenoid and the different type of carotenoid pigment attached to it. Okay, in this way. And these pigment molecules are called accessory pigment. Okay, now this type of a structure which is present here in the membrane of thylakoid is called photosystem, and this is photosystem 2. Okay, uh, this accessory pigment absorb light of a various wavelength. Okay, here I am showing you light. We know that light travel in waveform as well as a photon. Photons are the smallest particle of the light that carry energy. Okay, so different type of light energy is initially absorbed by this accessory pigments which is present in the photosystem too, and then this light energy is transported by every pigment towards chlorophyll A. Arrow showing the movement of energy from accessory pigment to essential pigment chlorophyll A. This chlorophyll A is called as reaction center. Okay, and chlorophyll A of photosystem 2 mainly absorb light of wavelength 680 nanometer. So this is called as P680. Okay, so this is the photosystem 2, which is the first photosystem that take part in the light reaction present in the thylakoid membrane, having accessory pigment and essential pigment. Essential pigment is a chlorophyll A. Accessory pigment absorb light of a different wavelength and then funnel the light energy or transfer the light energy by absorbing it to the chlorophyll A pigment. Chlorophyll A of the photosystem 2 mainly absorb or mainly use light of 680 wavelength. Okay, so we call this chlorophyll A as a P680, or overall the photosystems 2 can be reported as P680 or photosystem 680. Okay, now this chlorophyll A reaction center initially have 4 electrons. Suppose, okay, that means reaction center of the chlorophyll photosystem 2 contain de-energized ground state electron. That means these electrons are generally present at a ground state initially without energy. Okay, when light energy is transferred by accessory pigment to the photosystem reaction center, then the electron get excited. We know that when any electron is present in the ground state and absorb energy, it get excited and go into the higher energy level, and that energy level or the condition of the electron is called excited state. And due to the excitation of the electron by the light energy in the reaction center, this electron is moved or expelled from the reaction center. Okay, and now this 4 electron migrate from chlorophyll A to an enzyme called coenzyme Q, coenzyme quinone. Okay, this CoQ is the first electron acceptor in the light reaction. This CoQ accepts 4 electron from the reaction center, and this electron is generally moved due to light energy. Okay, when 4 electron reach to the coenzyme Q, they have high amount of energy. Okay, that means they are excited state electrons, and we know that electron cannot remain in excited for a longer period of time. They emit the energy and they again come into the ground state. Okay, and this process is occur by transferring the electron through electron carrier towards next photosystem, as I have shown here. Okay, now coenzyme Q transfer this 4 electron to another electron carrier, which is plastoquinone, PQ. Plastoquinone is the electron acceptor which accept electron from the photosystem 2 through coenzyme Q. Okay, generally we know that when we consider any electron transport system, there is a one molecule which function as electron donor, while other molecule function as electron acceptor. Okay, in this case photosystem to donate the electron, while plastoquinone accept electron. Due to donation of the electron, the space or vacancy is produced in the chlorophyll A reaction center, and this reaction center get ionized. Okay, that means the reaction center now get positive charge due to loss of electron. Okay, and now this electrons reaction center can act as a strong oxidizing agent. Okay, someone have to give electron to this vacant place so that the chlorophyll molecule is now ready for next cycle of electron transport. Okay, and this vacancy of the electron is filled by water molecule. This photosystem too lead to breakdown of water molecule. Water molecule is generally present in the lumen. Okay, this is the membrane of thylakoid and inside is lumen while outside is stroma. Okay, lumen contain water molecule and this photosystem too acts as a strong oxidizing agent and oxidize the water molecule. If 4 water molecule get oxidized, then 4 H plus is produced. They will going to enter in the reaction at the end. While after releasing 4H plus, what remain behind? 4HO. Okay, that means 4H2O molecule gets split. It is called as photolysis of water. Okay, and 4H plus and 4OH minus is produced. Now this 4OH minus give 4 electron. Okay, and due to release of the electron, 4OH minus convert into 4OH or 4HO. And now from this 4HO, one molecule of oxygen release, O2, in the form of gas, while uh, 2 molecule of water is produced. Okay, if you observe the reaction, this reaction carefully, you can easily balance it. 4H plus liberated now due to photolysis of water, while 4OH minus also produced. This OH minus release 4 electron and convert into 4 OH, and from this 1 molecule of oxygen is released. Okay, while 2 molecule of water is regenerated out of this 4. Now the 4 electron which is given by the water molecule transfer to the reaction center. Okay, that means the electron which is loose by photosystem 2 due to excitation of light is replaced by water molecule. Water play very important role in the light reaction, and that is electron donor. Okay, in this light reaction, water act as a main electron donor. You have to remember this thing. Now the first 4 electron which is transferred from CoQ to the plastoquinone. Now plastoquinone transfer that 4 electron to cytochrome B6. This is another cytochrome protein molecule, electron carrier, which is present in the membrane of thylakoid. And now from cytochrome B6, these 4 electrons are transferred to cytochrome F. Okay, and from cytochrome F, electron move towards plastocyanine. Okay, remember the names of electron carrier which is present in the thylakoid membrane. First is plastoquinone, then cytochrome B6, cytochrome F, and plastocyanine. Okay, when electron move from plastoquinone to cytochrome B6 and from cytochrome B6 to cytochrome F, they are releasing energy. So I have shown the path like this, because if I draw a scale of energy here, first electron going to the higher energy state and then they are releasing energy. So they are going downwards. Okay, de-energized electron or electron with a very less energy is accepted finally by plastocyanine. Okay, the energy which is released in the movement of the electron is used by cytochrome B6 and cytochrome F complex to pump hydrogen ion, as I said earlier, from stroma to the lumen. That means when electron move from cytochrome B6 to cytochrome F, hydrogen ions are transferred from stroma to the lumen. As electron flows, more and more hydrogen enter from stroma to the lumen of thylakoid, and number of hydrogen ion in the lumen increased as compared to stroma. Okay, so as we now know that stroma have less hydrogen as compared to the thylakoid lumen, so gradient is produced. Okay, and gradient always contain large amount of energy. This energy is used afterward for synthesis of ATP. I'm going to discuss about this at the end of this video. Okay, now once 4 electrons is transferred from photosystem 2 to plastocyanin, electron get de-energized. Someone again have to give energy to that electron and again transfer them from ground state to excited state, and this work is done by another photosystem which is present somewhere here, and the photosystem is photosystem 1 or P700. Okay, this photosystem is also similar with photosystem 2, having a reaction center which contain chlorophyll, then accessory molecules, accessory pigment molecules. Okay, this is photosystem 1, also called as P700. Why called as a P700? Because the reaction center of the photosystem 1 absorb light of a wavelength 700 nanometer. Okay, now the de-energized 4 electron is transferred by plastocyanin to photosystem 1 reaction center, and same thing happen again. Light of a wavelength, different wavelength, is absorbed by accessory pigments light energy, and then the light is transferred towards reaction center or funneled towards the reaction center by accessory pigments. And due to transfer of light by this accessory pigment to the reaction center, which is chlorophyll A molecule, this de-energized electron again get energized and transfer from ground state to excited state. And now the excited electron is accepted by a substance called pyridoxine reducing substance. Actually, this is not a well characterized compound which is present in the thylakoid membrane, but this compound accepts 4 electrons from photosystem 1, 4 energized electron from photosystem 1. Okay, and the compound is pyridoxine reducing substance. Okay, finally, the pyridoxine reducing substance give electron to pyridoxine FD. Okay, that when any substance accept electron, it get reduced. When 4 electron is accepted by pyridoxine, it get reduced. Okay, so this substance is helpful for reducing FD, so it is called as pyridoxine reducing substance. It play a role of reduction of pyridoxine, and finally from pyridoxine electron are transported, 4 electrons are transported to NADP. Okay, and due to 2 molecule of NADP get 4 electron, and then they convert into, they get ionized to form 2 NADP minus. Okay, each molecule of NADP get 2 electron from FD. So 2 molecule of NADP convert into 2 molecule of NADP minus, the ion form of the NADP, and now this ion form is ready to accept hydrogen ions. Okay, so these hydrogen ions which is released from the water molecule now come into the picture. These hydrogen ions is now accepted by 4 hydrogen ion is accepted by 2 NADP, and now 2 NADPH is produced. Okay, so this is the first product of the light reaction, non-cyclic light reaction. Electron leave from photosystem 2 directly transferred to NADP, and NADP convert into NADPH. Okay, so electron do not again come back in the photosystem 2 or photosystem 1. Okay, so it is called as non-cyclic flow of electrons. Okay, now this first product is required, we know that in the stroma for dark reaction approximately 12 NADPH is used by the dark reaction or Kelvin cycle for synthesis of 1 glucose molecule. Okay, this is the story of production of NADPH. Now what happened to the hydrogens which is accumulated in the lumen of the thylakoid membrane. Hydrogens are accumulated due to transfer of electron. They are pumped inside from stroma to the lumen of the thylakoid. Now this hydrogen is liberated. Okay, if I continue this membrane, if this is the membrane of thylakoid. Okay, now thylakoid membrane have a special enzyme hole, and that enzyme is called ATP synthase. This is the ion channel. Okay, this enzyme which is present in the thylakoid membrane is called ATP synthase. This enzyme having two part, FO part and F1 part. Okay, when high amount of hydrogen is accumulated in the lumen, hydrogen start moving out into the stroma, and when hydrogen from lumen pass from ATP synthase to in the stroma, energy is released, and that energy is used by this enzyme for production of ATP from ADP and inorganic phosphate. Okay, so the gradient which is produced inside the lumen is finally lead to the production of ATP, which is the second main product of the light reaction. Okay, so both the products are produced, NADPH and ATP, in the stroma, and stroma is the site of dark reaction. So they are now ready to enter into the dark reaction where they get utilized. Okay, now this is all about non-cyclic flow of the electron. What happens in cyclic flow? When plants do not require NADPH, or generally when plants have less supply of NADP, light is not available, Kelvin cycle is halted. Okay, if the Kelvin cycle is not in the function, in that case NADP will not be available for the non-cyclic flow of the electron. In that case, plant shift from non-cyclic to cyclic flow. To understand the cyclic flow, you have to just see this diagram carefully. When electron from photosystem 2 transferred to photosystem 1 and from photosystem 1 to FD or paradoxine, instead of going towards NADP, this electron again come back to cytochrome B6. Okay, so if you want to understand the cyclic reaction, just make one arrow from FD to cytochrome B6. Okay, and now you can understand cyclic flow. Cyclic flow only have photosystem 1. Okay, cyclic photophosphorylation do not require photosystem 2. No role of photosystem 2 in cyclic photophosphorylation. Only photosystem 1 play part. Okay, when light energy excite electron from the photosystem 1, they transfer to paradoxine reducing substance. From it, they are accepted by paradoxine, and from paradoxine they are again come back towards cytochrome B6, and from cytochrome B6 they transfer to cytochrome F, 4 electron, from cytochrome F to plastocyanin, and from plastocyanin to again photosystem 1. From photosystem 1 again light will going to excite them, they transfer towards FRS, FD, and this cycle continues. Okay, and this is called as cyclic flow. So what are the products of the cyclic flow? Cyclic photophosphorylation only produce ATP and not NADPH, because electrons are not transferred towards NADPH from FD, so this reaction do not occur. Okay, so NADPH is not produced in the cyclic flow. Electron, instead of going forward, electron come back from the FD towards cytochrome B6, and from cytochrome B6 to photosystem 1. In this reaction, hydrogen ions are pumped from stroma to the lumen, and then when concentration of hydrogen ion increased, they pass from ATP synthase, and then ATP synthase produce ATP. So cyclic photophosphorylation only lead to formation of ATP, but not NADPH. There are various differences which you can notice in between non-cyclic and cyclic flow. Like cyclic flow only involve photosystem 1 while non-cyclic have both photosystem 2 and photosystem 1. Then non-cyclic produce both NADPH and ATP, while cyclic produce only ATP. Non-cyclic photophosphorylation require water as the electron donor, while cyclic photophosphorylation recycle the electrons, so they do not require water or any other component as an electron donor. These are the some basic differences which present between non-cyclic and cyclic photophosphorylation. Here you can see photosystem to come in the picture or come in the process first and then photosystem 1. So why we do not call it as a photosystem 1 and photosystem 2? Actually, the name was given on the basis of their discovery. First, scientists discovered photosystem 1, and later on they realized that one more photosystem is present behind the photosystem 1, and so they have to give it as a name as a photosystem 2. Actually, process start from this point till we now know this system as a photosystem 2, which is P680, while the next photosystem is P700. Okay, so in this lecture, I discuss in detail about the two different type of electron flow which occur in the membrane of the thylakoid, and they are non-cyclic and cyclic photophosphorylation. Due to light reaction, light energy is used and it is converted into the ATP and NADPH2 or NADPH. ATP used as a energy reservoir or energy donor molecule in the dark reaction, while NADPH is used as a reducing energy carrier. Okay, and both these molecules are important in dark reaction. In my next lecture or next video, I am going to discuss about second part of the photosynthesis, which is dark reaction in detail. Okay, thank you.